Look at this lonely old macromolecule coming in, looking for some friends in its good old protein setting.
Hey, can I sit with you? Look at you, you're wearing pink. Today's not Wednesday. On Wednesdays we wear pink. So no, you cannot sit with us.
Like many of the...
I mean, I haven't even washed any of my clothes, but that ain't no big deal.
Bye.
Like many of the receptors, only certain types of post-translational modification will be compatible in cells to recognize each other. And here you can see her meet her own friends.
Hey, what's up, y'all? Can I sit with you guys? Because the other group was being a little weird and rude, so I need to make some new friends.
Oh my god, yes! Let's fucking make a sandwich! Oh my god!
And now they're all happy, being post-translational and compatible and a successful cell. Now, glycosylation is a type of post-translational modification on proteins. To understand it, it is important to know that this word possesses the first part, which contains glyco, meaning glucose. This underlines the fact that all of these glycosylating modifications involve sugar additions. There are many different types, which include N-linked, in which the glycan binds to the aminograph of the asparagine in the endoplasmic reticulum, which is the most common, but is also extremely diverse. The next type is O-linked, in which the monosaccharides bind to the hydroxyl group of serine or threonine, the ER, the Golgi, cytosol, and nucleus. The next is glycation, in which the glycan core links a phospholipid and a protein. In C-linked, mannose binds the indole ring of tryptophan, and in phosphoglycosylation, glycan binds to serine via phosphodiester bond. Now, a common application for this is the recognition of other cellular factors. Specifically, in our red blood cells, glycosylation is utilized to give our cells our different blood types. Now, by now you probably know that there are A blood types, B blood types, AB, and O blood types. Regardless, the minus factor, which is dependent on the rhesus factor. This is all determined by the fact that these cells are glycosylated with specific proteins, as shown here, which give different shapes for the antigens to be recognized by the cell and give themselves specific properties.
Phosphorylation regulates protein function in which transmits the signals through the cell. It is the most common mechanism in which adds one or more phosphate groups to an organic compound. Phosphorylation is one of the most important and well-studied post-translational modifications, and it plays a significant role in cellular processes in eukaryotic cells. In this general diagram, ATP, which is the energy carrier of the cell, transfers a phosphate group in the side chain of the protein. This catalyzation involves a reaction by a protein called kinase. The addition of this phosphate group signals activation or deactivation. This picture only shows the activation of the cell by the addition of the phosphate group. Although not shown here, the addition of a phosphate group can also deactivate the cell. A second enzyme known as phosphatase catalyzes the removal of a phosphate group. It is important to know that adding a phosphate group can either increase or decrease the activity of the protein, varying where the site of phosphorylation occurs. While phosphorylation is a prevalent post-translational modification for regulating protein function, it only occurs at the side chains of 3 amino acids, serine, threonine, and tyrosine in eukaryotic cells. In this example, ATP is used to add a phosphate group from the enzyme kinase. The addition of this phosphate occurs at the side chain of an amino acid called serine. Shown in pink is a phosphate group at the side chain of serine, which is now called phosphosirine.
And the hydrogen bond gets taken away, and the carboxylic acid right here is being added. So where it's added is right here. It takes away this piece right here, it adds it, and then it breaks the last piece right here and adds the hydrogen atom. Now you may ask, why is it important for this chemical reaction in prokaryotic cells as well as eukaryotic cells? Now, I'll tell you why. Because the prokaryotes have the capacity to acetate both the alpha amino groups as well as the beta amino groups in the N terminal. And many eukaryotic proteins are modified by differential internal acetylation, which is very important for a variety of processes. Now you might wonder which processes. Now this is crucial for DNA replication, for metabolism, as well as gene regulation. So now you know more about protein acetylation.
Ubiquitination is a post-translational protein modification that is found exclusively on eukaryotic cells, meaning that they are not found at all in prokaryotic cells. Ubiquitination is a process in which multiple ubiquitin molecules are attached to a target protein that needs to be broken down. But before I begin talking about the whole process of ubiquitination, I believe it is crucial for me to address what a ubiquitin is. A ubiquitin is a 76 amino acid protein that is used to mark proteins for degradation. So now I'm going to begin talking about the whole process of ubiquitination. Ubiquitination requires 3 different enzymes and 3 different steps in order to occur in the body. The first step is the ubiquitin activating enzyme, E1, and this step is done through the hydrolysis of ATP. The carboxyl end of the ubiquitin is linked to the enzyme through a thioester bond. And then in the second step, ubiquitin conjugating enzyme E2, the ubiquitin enzyme from step 1 is then transferred into the second enzyme by attaching to the cytosine residue of E2. And then in the last step of ubiquitination, ubiquitin protein ligase, E3, this enzyme shuttles the ubiquitin to the target protein. And then in the last step, this is really the one step in which they are attached to the specific targeted protein that needs to be broken down. This whole process occurs a minimum of 4 different times in every different targeted protein, and the way that they are able to attach to each other is through isopeptide bonds. And this whole step is reversible through the process of de-ubiquitination, but this is literally the whole process.
Amino acid degradation is a process of deamination reaction. Deamination is a process where an amino group are removed from a molecule or the conversion of an amino group. So deamination is more or less the breakdown of amino acid. Deamination has 3 main different reactions: oxidative, hydrolytic, and eliminative. Oxidative is a process where an amino acid loses its animated functional group and is then converted to a keto acid. Hydrolytic deamination, water is added to the amino group. The amino group gains H2O but loses ammonium. Deamination is the process where hydrogen or sulfide is removed from the amino acid. This process usually only occurs in small amino acids. Forward reaction versus reverse reaction. In forward reaction, oxidative is the formation of deamination, the structure of alpha-keto acids. This is an aerobic process of deamination that usually takes place in the liver or kidneys. In reverse reaction, reductive amination is the formation of amino acids. So reductive aminations form carboxyl groups to enamine.
Proteolysis, the breakdown or degradation of proteins through the hydrolysis of their peptide bonds. Proteins are broken down in the reaction, resulting in peptides and sometimes amino acids. The reaction requires protease, an enzyme which can be found in prokaryotes and eukaryotes in both the nucleus or cytoplasm respectively. This is a proteasome, a protein complex which contains protease. The proteasome is made of 2 parts, a core in which the reaction takes place, and 2 regulators which serve almost like caps or doors for the reaction, allowing it to only degrade the necessary proteins. It works with ubiquitin, another enzyme which signals which protein needs degrading. Proteins with the ubiquitin attached to it approach this complex, and the ubiquitin binds to these regulators, almost like a key opening a door, activating the proteasome, letting the proteins in and enabling proteolysis to take place. Once the reaction has completed, the remnants of said protein exit through the other side as peptides.